Hello, everyone. Good afternoon from Zurich. My name is Maria Bien. I am your host for today, and I'm very happy to introduce our invited speaker, Professor Hideaki Yamamoto, who is currently in Tohoku, Sendai, Japan. So, hello, Yamamoto Sensei. So our agenda for today is as follows. We will start with an introduction from Asobai Systems by myself, and then we will proceed with our main presentation for today's webinar, which is by Professor Hideaki Yamamoto. Then we'll have the Q&A session in the end. So don't hesitate to communicate with us through the chat, but please add your questions in the Q&A, otherwise we will not be able to answer them. All right, so let me then proceed to the first part, which is the introduction. So our presentation for today is about from cells to algorithm, next generation high density microelectrode array as a biocomputing platform. So if you don't know yet, our platforms at Muscle Biosystems are as follows. So we have the Max1 system, which is our flexible platform for our single well high density microelectrode array. It's super flexible because you can use it for any type of experiment, and it's also very small. You can put it inside of the incubator, which is especially important for biocomputing since you want to do longitudinal experiments and recordings. Our Max2 is the counterpart for high throughput experiments, and it is compatible with 624 well plates and for our upcoming 96 well plate. So if you want to learn more about our systems, please go ahead and visit our website. But today, I would like to focus more about the biocomputer. So what are the components of a biocomputer? Of course, we would need biological samples, whether that is 2D cultures or organoids. And then we need to have proper culturing techniques and also maintenance of these cultures. Of course, if you would want to have more complex networks, which we will also see today in the presentation, it would also be good to indicate or to include some microstructures using PDMS or other devices. And finally, to have a biocomputer interface. In this case, that will be our system, our high-density microelectrode array. So what are the needs for both in vitro electrophysiology and for biocomputing? So the first part is precise and reliable functional readout. Here at Muscle Systems, we are very proud to offer low noise and single cell resolution for our high density MEAs. And for today, I'm also excited to share with you some of our new developments to enable even further the versatility for acute and longitudinal recording of any sample type. Here you can see examples of recordings from different types of organoids that are mostly used as well for biocomputing. Now, more about our HD-MEAs. So this is how it looks like. This is our single well system. If you zoom in, you will see a 2 by 4 millimeter square area filled in with 26,400 low noise electrodes. And any of these electrodes can also be used for electrical stimulation. So you can play the piano with your cells and be flexible enough to do any sorts of feedback for your cells. The same chip is being used for our multi-well, which is also for the 6 well and for the 24 well. So you just increase the throughput while having the same capabilities. Now, to enable better for your experiments, we have two accessories that we would like to share with you today. One is what we call the dot plating insert. So what it is essentially a tool to be able to allow us to center the cells properly and always have the same cell density independent of who it is that is doing the plating. So here you can see an example of how the 24 well plate dot plating insert is being used, and you will see that it's going to be very easy. There's a guide for your pipette, and you can also do it quite quickly without needing to really center and look at the center of the array. So here you can see how the dot placement looks like for the one with the device and for the one without the device. Of course, also for without the device, there's a risk that while you're moving your well plate or your chip, that this dot can also move around when you're coming from the hood to the incubator. So if you're interested, please contact us, and it's already available now. Next, for organoids or 3D samples, you would want to have both stability and tissue health. So for normal conventional devices, this is usually like a rigid grid that is just sitting on top of your organoid, which has a fixed geometry, compression-based, and it could flatten the tissue and is also sensitive to the size and the shape of the organoid. Here at Muscle Systems, we would like to support any types of experiments that you would like to do with organoids. So for acute recordings, we also have now the Max 1 organoid holder. So it has an adaptive membrane that is like these flexible fingers that softly goes on top of your organoid. It's non-invasive, not altering the shape of the tissue. It fits diverse organoid shapes. And also you can recover the tissue after the recording, put it back in the incubator, and then record again the next days. So it looks like this, how it works. So you can center your cell or your organoid, and then you can just place the holder on top of it with the help of the handle. So it's quite easy to use and is currently available for the Max 1. Okay, so those are the new accessories that we have. I hope you're excited about this as we are at Maxwell. The second need for in vitro electrophysiology and biocomputing is an ideal surface for microstructures. And here, our Plus chip, our Max 1 Plus chip, has a 0.3 micrometer surface topography that allows us to place really nice structures and make sure that all of the axonal projections are just within the structure that you have, similar to what you can see here in this figure. So what you can do with this device is, for example, connect multiple organoids or connect multiple cell cultures together, and then be able to record them using our high-density microelectrode array. Okay, the third one is flexible stimulation and control. Here, of course, we know that there are two ways to be able to do biocomputing. One is an open loop approach, wherein you can use the organoid or the cell as a reservoir. And the next one is by having some feedback loop with a closed loop approach, which is also used by Kagan et al. In their Pong paper. And how we are enabling this at MuscleBuy Systems is that we have the MASAB Live API that allows you to communicate with high-density MEA hardware and also stream the data live and be able to interact with it with any way that you can. So if you want, again, to learn more information, please come and visit our website or contact us. And finally, I think this is also quite important aside from the tools, the cells, and the equipment. An open and collaborative community is also good to, progress in the field of biocomputing. And we are very, very happy today to introduce our webinar, which is focused on neurocomputing. This year, we really have a lot of focus on this topic, and we have a lot of tutorials, blog posts, webinars like today, and user perspectives. So if you're interested, please come to our website, scan this, and explore our resources. That's it from our side. Now let me proceed to our invited speaker for today. I would like to introduce Hideaki Yamamoto. He's a professor at the Research Institute of Electrical Communication at Tohoku University, Sendai, Japan. He obtained his PhD degree in engineering from Waseda University in Japan in 2009. He was also a JSPS Research Fellow at Tokyo University of Agriculture and Technology and an assistant professor at Waseda University before joining Tohoku University in 2014. In 2020, he was appointed associate professor at the RIEC and was promoted to professor in 2026. Congratulations, Yamamoto-sensei, for your promotion this year. So his research interests include the use of engineered neuronal cultures as a model system for understanding brain computation, as well as their applications to brain-inspired computing and biomedicine. So it is our honor, and please help me welcome our guest speaker for today, Professor Hideaki Yamamoto. The floor is yours now.
Yes. Right. So Marie, thank you very much for the introduction and also for the invitation to present our work. I'm Hideaki Yamamoto, and I'll talk about this topic. And I first want to start off by mentioning that this work is a collaboration with multiple scientists. I especially want to mention Professor Katori at Future University of Hakodate, and Yuki, Yusei, Professor Hirano, Professor Sato at Tohoku University, and Takuma and Yuya at the University of Calgary and Waseda University for the collaboration on this. So my main appointment is at an institute called the Research Institute of Electrical Communication that specializes in information and communication technologies. And in my group, most of the members, including myself, have a background in electrical engineering. But still, we share a strong admiration towards neuroscience, And we use these semiconductor clean room facilities here at the laboratory for nanoelectronics and spintronics to fabricate homemade microfluidic devices or microstamps that we use in experiments and also share with our collaborators. And what we do with these tools is that we take control over the growth of living neurons in a petri dish to build in vitro models that capture the essence of complex in vivo networks. And today I'll first start by describing the background of our research, what we want to do at the interface of neuroscience and microfabrication. And then I'll describe some of our recent works on the in vitro reconstitution of neuronal networks possessing modular organization. And secondly, reservoir computing approaches to decode neuronal dynamics of these engineered neural networks. And then I'll finally comment on some of our recent attempts to bridge top-down and bottom-up approaches towards the end. So the fundamental question that my group is interested in is the question of how do complex brain functions arise from the collective activity of neurons? The neurons that comprise our brain is essentially an unstable chemical element, and it's basically a simple integrate and fire unit. Yet its network somehow performs various complex functions as a brain autonomously and adaptively using these biological elements as its physical substrate. And in neuroscience, most common way of studying this relationship between the neurons and the brain is the so-called top-down approach, where we try to understand the working principles by analyzing the system in detail. And this kind of top-down investigation is playing critical roles in neuroscience, but on the other hand, as a property of complex systems in general, when we try to understand this relationship between the functional element in a system, not only such a top-down approach, but also an inverse, a bottom-up approach is necessary, in which we try to understand the system by building it from its elements. And here, one of the important tools in studying the brain in this top-down manner is the technology of growing the neurons in dishes. But when we culture neurons in dishes, the neurons tend to connect randomly with each other, and the network starts to produce a strong bursting activity that is excessively synchronized across the network. And so there's a fairly large gap between the in vivo and in vitro systems, both in terms of their connectivity and the activity that they produce. And my motivation has been to somehow bring these two systems closer using cell engineering technologies based on microfabrication that we specialize in. And manipulating these culture neurons using engineering methods is not something that came up recently, and pioneering works date back over 30 years. And for instance, a group in MTT Labs first used substrates with microgrooves to orient the direction of neural outgrowth of primary DRG neurons in culture back in 1988. And nearly at the same time, a group in Bell Labs first showed that a pattern of aminosilanes and alkylsilanes can be used to define the adhesion sites of cerebellar neurons in culture. And these technology has then matured over time. For instance, this is a micrograph from a paper by Professor Offenhuiser's group in Jülich in 2005. Here you can see that neurons are aligned against discrete micropatterns at a single cell resolution. And here in the same paper, synaptic plasticity, which is the basic form of learning in neural networks, has been shown in these engineered neural networks for the first time. And more recently, there's also been a demonstration of a nanoscale patterning of subcellular structures like dendritic spines, and controlling the directionality of neuronal connection is a topic that has attracted engineers for quite some time. And in our group, we recently showed that by implementing these directional connections in the modular neural networks that I'll talk about later, this helps suppress. We found that this helps suppress the excessive synchrony and enrich the dynamic patterns embedded in these networks. And of course, the field has also advanced through the incorporation of iPSC-derived human neurons and organoids to reconstitute biological neural networks with human cells. And meanwhile, while all this was happening, a number of breakthroughs were made in the surrounding fields that amplified the scientific impact of the cell engineering technology. And in my opinion, the most important of all is the graph theory in applied mathematics. And this is a very old discipline that dates back to Euler, but after the seminal work by Watts and Strogatz in 1998, the field has expanded to provide theoretical frameworks for analyzing complex real-world networks like social networks, internets, and of course, the brain networks. And another field that I need to point out is the rise of connectomics studies in neuroscience, which aims to obtain a comprehensive mapping of neural connections within organisms' nervous systems. And driven by large-scale funding, such as the Brain Initiative in the US, anatomical information of the brain networks is now beginning to become available at a cellular resolution. And based on these work and experiments, one of the canonical network structure that were provided is what's called the modular structure that I'll talk about today. Modular networks are characterized by the presence of multiple densely subnetworks, which are called modules, that are sparsely interconnected with each other. And shown here are matrix representations of neural connectivity in the nervous systems of various species, ranging from C. elegans to humans. And colors here indicate the strength of connections between neurons or neuronal regions. And as you can see here, the cells or the brain regions are not randomly nor uniformly wired, but has the structure where they form a group which is more densely coupled within each other than to those in other groups. And so in the remaining slides, I will go over some of our recent studies showing how neuronal networks cultured in microfabricated substrates serves as a bottom-up platform to understand functional roles of such modular organization, and show how high-density MEAs enable closed-loop coupling between electronics and biology. And there are multiple methods for patterning neuronal networks, but the most popular approach now is to use what's called the microfluidic devices made with PDMS. And we fabricate our own devices using the clean room facility at our institute. The fabrication starts with the fabrication of the master mold. We do this by photolithography, first by patterning a thin layer of SU8 to make the microchannels, and then secondly with a thicker SU8 to create the through holes or the wells where the neurons go in. And we then drop gas PDMS gel onto this mold, harden it by thermal curing, and then peel off the PDMS to obtain the microfluidic device that looks like this. And one of the unique features of our microfluidic device is that we made this device thin so that the through holes where the cells enters the device are there when we peel off the PDMS from this mold, and that there's no need to manually punch out the through holes using tools like biopsy punches. And the pattern design looks like this. It has multiple square wells that are connected by thin microchannels. The square wells are typically 100 micrometers or 200 micrometers on a side, and lets multiple neurons go in and form dense connections within each well. And the microtunnels are several microns in height and width, which are too narrow for the cell bodies to go in, but it lets the axons and dendrites to penetrate into them. And this allows controlled coupling between the neighboring wells. And these devices hence allows us to create multiple densely coupled subnetworks that are sparsely interacting and therefore a modular network. And for today, I'll skip further graph theoretical description of this, but by growing rat cortical neurons in these devices, we showed that excessive synchrony can be suppressed, balancing the synchronous and asynchronous activity in these networks. And such a balance of synchrony and asynchrony, or the integration and segregation, is firstly an important portrait of neural activity in animal brains, but also plays an important role when using these systems in reservoir computing tasks that I'll come back to later. And while we have been using calcium imaging as the primary technology to record neural activity, we were very lucky to be able to introduce the MAX-1 system in our group in 2019 and see that such activity can be captured by MEAs. And these are spontaneous activity recordings. Maybe I should come back to and play it again. Activity recordings from rat cortical neurons patterned in 2x2 modular configuration. And here the patterns were created by microfluidic devices that were placed on top of these chips. And the data here was taken by Yuya Sato, who is now at Waseda University. And what is really cool here is that high-density MEAs allowed us to capture not only the propagation of activity across separate modules, but also the propagation within individual modules that occur in sub-millisecond timescales. And these were something that we previously could not resolve by calcium imaging. And another important feature of the MEAs is that, as Marie mentioned, that the electrodes can be configured to stimulate these neurons. And taking advantage of this feature, we can see that diverse activity patterns can be evoked by stimulating different sites of individual networks, leading us to consider using these systems for reservoir computing. And more recently, we started taking advantage of the large electrode area of the MAX-1 chip, which is approximately 2 millimeters by 4 millimeters, to create large-scale networks. And here we're showing networks that comprises 128 modules and contain nearly around 2,000 neurons. And you can see that when the neurons are grown without the microfluidic device, the cells generate the so-called network bursts, where the neurons fire simultaneously, which are separated by relatively quiescent periods. In contrast, in the patterned networks, they produce more diverse activity patterns that involve a mixture of synchronous and asynchronous events, as I showed in the calcium imaging data. And what's important is that in these networks, if you look at the range of the vertical axis, which shows the mean firing rate, even when the neurons are firing together collectively, the amplitude of the mean firing rate is either half or even a quarter of what was observed in the homogeneous case. And one way of quantifying the richness of the activity patterns is to do a PCA on the network dynamics and look at the distribution of the explained variance. And here you can see that in the homogeneous network, the first principal component, which more or less corresponds to the entire network activating together, already explains 70% of the variance, and that the pattern in the pattern networks, they require a larger number of components to explain their dynamics. In other words, these engineered networks exhibit higher dimensional dynamics. So to make a short summary up to this point, what we're seeing here is not exactly the same as what scientists observe in vivo, but we're beginning to be able to bridge the in vitro networks closer to the in vivo networks by using cell engineering technologies. So, so far, we've been looking at the spontaneous activity of the network, and spontaneous activity is an important portrait of information processing in the brain, but there's more to it in the brain. And as Dr. Yuri Buzaki noted in his book, nothing in the brain makes sense except in the light of behavior. And studying behavior in cultured cells is a bit tricky, but we can at least stimulate the cells and see their responses. And so, and we started building setup to let biological neurons interact with artificial environments, and at the same time look for ways to interpret the evoked activity patterns from the perspectives of computing. And the framework that we use to do this is reservoir computing. And reservoir computing was first proposed sort of as a lightweight variant of recurrent neural networks. And in reservoir computing, the output weights here are trained while keeping the weights of the reservoir layer fixed, which substantially reduces the computational cost for training. And in order for a dynamical system to work properly as the reservoir layer, they have to possess several characteristics such as the nonlinearity, echo state property, or the short-term memory. But once several conditions are met, an arbitrary system can be used as the reservoir layer to replace the recurrent neural networks. And cultured neurons and more recently brain organoids have been shown to actually possess such properties to some extent and have been used successfully as the reservoir layer for certain tasks. And this kind of application was first pioneered by Dr. Tom DeMars, who demonstrated the separation property in cultured neural networks back in 2009. Sadly, he passed away far too young 5 years ago, but it's a great memory that I had a chance to host this visit back in 2015 and discuss science with them in person. And later, Takahashi Sensei's group in Tokyo first demonstrated an online supervised training of such biological neural networks. And our first work in this field appeared in 2023. And from around the same time, we've seen a growing number of publications from groups all around the world mirroring the rising interest in the field of reservoir computing and also in biologically inspired AI. And what we did in our recent paper is to implement the so-called time series generation task in the cultured neurons growing on the MAX1 chip. We take a cultured neural network, record its activity using MAX1, and decode an output using a linear decoder to produce the desired time series signal. And based on this output, we deliver a feedback stimulation to the neurons and ask whether the readout weights here can be trained in an online manner so that the cultured neurons that are driven by its own spontaneous activity and this feedback from the output can generate designed time series signal, such as the sinusoidal wave or chaotic attractors. And Maxwell offers a Python API and a C++ script to interface the recordings with the external computation. And we took advantage of this to set up this system. And this is a representative result of what we have achieved. And this figure shows the filtered activity patterns recorded in 10 representative electrodes, along with a target signal, in this case, a sinusoidal wave with a 30-second cycle in gray, and a decoded output is shown in red. First, when we record the spontaneous activity without any closed loop feedback stimulation, the network in the patterned modular configuration exhibits a variable activity pattern with a certain amount of synchrony, as we saw before. And once we close the loop and switch on the training, the activity patterns become more periodic and structured in synchrony with the target signal, with a certain electrodes active during the positive phase of the target signal, and the others active in the negative phase of the target signal. And at this stage, you can see that the red trace, the system is able to output a signal that more or less follows the target signal. And after certain steps of training, the changes in the readout weight somewhat converges. And at this point, the weight update can be turned off. And in successful cases, not always, the system can self-sustain the signal generation. And if we do a PCA for dimensionality reduction and plot the trajectory of the activity patterns, we can see that the spontaneous activity wanders around a broad high-dimensional space region of the state space, whereas under the closed-loop feedback, the activity is more clustered, and goes back and forth between two states that more or less seem to match with the positive and negative peaks of the target signal. The feedback control shifts the BNN, the biological neural networks, from a highly variable to a structured state, and engineered biological neural networks can be trained to generate such periodic signals. And this can be applied to other types of periodic signals, like a triangular wave or a square wave. But we can see that at the time points that require high frequency components, those points were more difficult to follow, which could partly be due to the filtering that we apply to the recorded spec trains. And what was important in our work is that the engineered networks shown in blue and orange perform significantly better than the homogeneous networks that were grown without the microfluidic patterning. And this points to the importance of neuroengineering technologies in making these line of research possible. And we also found that an identical network can be trained to self-sustain sinusoidal signals with different periods, pointing to the adaptability of biological neural networks. And finally, on the right, here we asked what makes a good biological reservoir. And there must be multiple factors that come into play, but one parameter that we looked into was the dimensionality of the network dynamics, which we quantified using a measure called the effective rank. And this essentially quantifies how many principal components are meaningfully active to explain the observed dynamics. And plotting the mean squared error against the effective rank across all samples gave an inverse relationship, pointing to one of the mechanisms behind more accurate regression with these biological reservoirs, meaning that higher dimensional reservoirs exhibit higher performance. So to put this result in a broader context, training recurrent neural networks online to generate temporal signals was first demonstrated by Sosilo and Abbott in 2009 using rate neuron models, and later extended to spiking neural network models by Nicola and Kolpath in 2017. And what we showed in our recent paper is that similar computation can now be implemented in biological neurons as well. So my point today is that a combination of cell engineering and high-density MEAs gives us a way to better understand the function of neural networks in the so-called bottom-up approach, where we try to understand the system by building it from the elements. And today I showed that it can be used to understand several aspects of modular organization that they promote integration segregation balance and also supports online supervised learning to generate temporal signals. And here, high-density MEAs enable closed-loop coupling between electronics and biology. So these tools are useful, but on the other hand, if we want to extend these observations to the understanding of our brains, in vitro experiments are not sufficient by themselves. And here we need a strong collaboration between in vivo experimentalists studying the brain from top down and theoreticians that develop models that unifies the findings from both directions. And this motivated us to launch a new research consortium that we termed Multicellular Neurobioncomputing, which is funded by the Grant in Aid for Transformative Research Areas aid program in Japan. And we're a team of 42 PIs and co-investigators from 21 institutions in variety of fields ranging from information science, bioengineering, neuroscience, and electronics. And we're working together to combine top-down and bottom-up approaches to better understand the principles of biological computing based on multicellular networks and developing systems applications. And when we gather researchers from such diverse fields, one thing we try to foster is the sharing of cutting-edge resources among members so that all the members can access state-of-the-art equipment, even in areas that they don't necessarily specialize in by themselves. And a natural example of this was the Max-1 system, which was installed in Professor Tani's group at Waseda University. And this has been recently used by Nishimura-san at Doshisha University. He's a stem cell biologist, and he placed his human striatal and midbrain assembloid on the Max-1 at Waseda University and recorded its activity. And this work was recently posted on bioRxiv and is now under review in a paper. And I would also like to mention that the reservoir computing experiment that I talked about today was also a collaboration with Katori Sensei, who is one of the PIs of this project. And we're almost at the midpoint of this program, and please stay tuned for more to come. So before wrapping up, I would like to thank the Maxwell team for their continuous technical support, and in particular for this closed loop experiments. This goes especially to Dr. Robert Giglietto, Dr. Ed Lee, and Dr. Jan Miller, and their support was inevitable to make this work possible. And I would like to thank all my collaborators, especially Yuki and Yusei and Professor Katori and Yuya and Takuma, and also the fundings. And with that, I would like to end, and I'm happy to take any questions, if any.
Thank you so much, Yamamoto Sensei. Really, really, really amazing results. And just watching the presentation really gave me goosebumps. Thank you so much for sharing this with all of us here in the audience. And also, we are very, very happy to be part of your work. Thank you so much for having Muscle By Systems be part of this really great research that you're doing there. Okay, first question is from Nina. Hello, Nina. So she asks, in the homogeneous versus the 128 node neural networks, are the total amount of neurons approximately the same in random versus modular conditions? Could there be an effect of the network size in the dimensionality of the dynamics?
Good point. So we plate the neurons at the same concentration, and so in that case, the neurons, the number of neurons should more or less be the same. But since there's an effect of the PDMS inhibiting the adhesion to those regions, I could suspect that the homogeneous networks have a larger number of neurons compared to the patterned networks. And so the second half of the question was, could there be an effect of network size on the dimensionality of the dynamics? There could be, but we couldn't. What we can interpret is that since there are a greater number of neurons in the homogeneous case, the reduced dimensionality in the homogeneous case is not attributed to the effect of reduced number.
Thank you so much. Next, we have a question from an anonymous attendee. Why do you think that higher dimensional reservoirs exhibit higher performance in the sinusoidal wave generation experiments, even though when you plot the PCA components in the activity post-learning, there are low dimensional representatives representing the peaks and the valleys of the wave?
Right. So in the reservoir computing, it's not always true that higher dimensionality positively correlates with higher performance. So if you have a dynamics that is too high in dimension, it could be too noisy to be used as a reservoir. But in this case, I guess what's happening is that that we're sort of looking at the low dimensionality regime where the higher dimensionality acted positively. So, that's a good point. And whether higher dimension, this high dimensionality helps should also depend on the task itself. There must be a task dependence. So that's a very interesting point, and we should look more into it.
Thank you so much. We are now proceeding to the next one. So according to Han, thank you for your talk. What other methods are available to prevent the highly synchronized burst activity in 2D cultures and organoids? This is a curious question.
Well, yeah, so I mean, you can do it pharmacologically if you block excitatory neurotransmission. Let's say not fully, but partially, it could reduce the synchronized bursting, for instance. You could, I think, you could plate the cells with lower dimension, lower density, and it would reduce the connectivity between the neurons. But in any case, you would have to end up in a sort of pharmacologically perturbed state or networks with very low density that eventually can form a dense network to produce synchrony. So in that context, I think these patterning technologies are useful in reducing the synchrony.
Thank you. The next question is from Michael. How durable are the connections between the HD-MEA surface and the neurons, and how well do they adhere?
Well, so for this particular paper that we worked on, Yuki has been culturing the cells for up to 1 month, and it's been stable, especially with the flatter Maximum Plus chip that Marie described in the first half of this webinar. So at least 1 month they work. I don't really have the number beyond that, but I think there are many more people working, especially in human cells that are doing longer term cultures.
Thank you so much. And if you would like to learn more about how to adhere cells on our HD-MEAs, we do have cell culture protocols available online. And I think we also have some application notes that show over 3 months or even 6 months of cultures already well attached still. So thank you for the question. All right, next we have Stephen. Thank you. Have you explored different numbers or sizes of compartments in the modular structure, or having some asymmetry in sizes across the lattice?
No, as of now, and the only structural changes that we've explored are the so-called lattice network and the hierarchical network that I sort of described. Maybe I should come back to this slide. Comparison of these two configurations that have a different pattern of connectivity between these wells are the only comparisons that we've done in structure-wise, and of course, we initially expected that these hierarchical networks perform better in these reservoir computing tasks, but it turned out that the lattice networks produce higher task performance when we compared the MSEs, the mean squared error between the target signal and the actual output in any of these cycles.
Mhm.
And we interpreted this as that that in the lattice networks they were less asynchronous, but the activity, the firing rate of in the neurons were higher compared to the modular, in this case, the hierarchically modular networks. And our current understanding is that this desynchronization combined with higher firing rate is helping the higher performance in these in the tasks that we explored in this paper. But again, as I mentioned in a previous question, it could be task dependent. There could be tasks that where the hierarchical structure perform better than the lattice networks.
Very interesting. Thank you so much for the answer and for the question. Before the next question, I will go back to Jonathan, who now clarified what his question is about. So he was curious, how does the input stimulation pattern look like to produce the result, the sinusoidal wave or the other waveforms?
So that's a very good point that I skipped. The input is basically a biphasic pulse that people typically use in these MEA experiments. And what we did was to map the amplitude of the sinusoidal wave to the amplitude of the biphasic pulse that it gets applied.
And you applied it on all the modules.
Simultaneously, we could stimulate up to 32 neurons at the same, 32 electrodes at the same time, but we restricted to, just for safety, 30 electrodes. And so the stimulation is delivered to 30 randomly selected electrodes.
Thank you so much. I hope that answers your question, Jonathan. Next, we have Joel. Hello, Joel. First, thanks for the talk on slide 22. Okay, maybe we need to go to slide 22 now, Yamamoto Sensei. You mentioned to train the biological neural network. The way I understand the system is that you solely train the in silico readout layer, but not the other aspects. Did I miss something?
No, you're absolutely correct. It's the output layer that are being trained, and there are works that looks at training that occurs within the biological neurons. But in my study, we're looking at we're treating the biological neurons as a fixed reservoir layer.
Thank you so much. Next, we have a question from Kalia. Hello, do you have any recommendations for choosing filters? I am getting started in this kind of work.
It depends on which filters is being mentioned. So if it's the filtering that I apply on the spike trains, this is an important important aspect, and is also depends on what kind of task that you want to do. And for our case, we wanted to create a sort of a very slow temporal signal. So the filter was the double exponential filter that I used was set to a relatively large time constant.
Thank you so much. I guess depending on the question or the task, this can then be adjusted accordingly.
Yes.
Next question, Vias. Hello. Does the geometric organizations of neurons matter? I mean, the nervous system of all animals are 3D.
It should. It should. Um, so I guess we're looking at what we can do with 2D
Yeah.
as of now, and going onto 3D is of course interesting, but
Mhm.
there are a lot of challenges in terms of culture and also the recording that needs to be done, and I think both ways are important. Looking at what we can achieve or what we can understand with 2D, and also simultaneously developing technologies for 3D culture and 3D recordings.
Thank you so much. Just information for all of our audience. I see that we have a lot of questions in store for us here, but we only have around 4 minutes left. So just in case we're not able to capture all of your questions, we will answer them online by email. So don't worry, don't worry. Just keep on asking your questions, and we will try to go through them one by one. Okay, next question by Juan. How long do the networks retain the learned behavior after the training session? I'm actually curious about that. In closed-loop experiments, the learned response often fades shortly after training, sometimes within just minutes. And great talk.
Thank you very much. And as I mentioned in my talk, and also in the paper, whether the free running mode, what we call the free running mode, where we turn off the weight updates and lets the activity to self-sustain. This is still tricky, and it's not always that they work. And so I would not, you know, uh, like to make a strong comment on how long it can sustain. But in successful cases, we have shown that they can sustain for 300 seconds at max. But letting this free running mode work is still a challenge for letting this free running mode work stably is still a challenge for us, and we're still looking into that.
Thank you. Next, we have a question from Frank. In hierarchical modular networks, are the connections between the squares directional, or can neurites grow bidirectional?
So we are working on making asymmetric connections, but that was in a different project. And for this particular project, we're always using symmetric connections.
Thank you.
So they're bidirectional.
Thanks a lot. Next question from Ramez Daniel. How does latency in the closed feedback loop affect the learning process? Is there an optimal latency range?
So we looked at this in computational simulations using spiking neural networks, and there it was our results were that lower latency tend to produce a higher performance. So I think it's a technical challenge for us now to reduce the latency and see if the performance improves. It's a very, very important point.
Thank you. Thank you for the question. Um, okay, next, we have Michael again. Would the neurons be dislodged from their intended electrode position if the HDMA were to be bumped or jostled around beyond the standard lab environment conditions? I guess if you move around the chip, will the cells move around as well? Was the question.
I don't know. Does it?
I think the modules actually protect them quite a bit from any jostling of the media. So
I see. If I just comment on as far as what I know,
Mhm.
so we do the activity scans every day. Even if we come back to if we use the same chip, the same culture for this experiment in the next day, we do the activity scans where we look for active electrodes on that day. So that would prevent the movement of the cells or the neurites with respect to the electrode patterns.
Thank you so much. I think we just have one more question remaining. Here I'm going for Florian. He said, thanks for the talk. Is there a possibility that the neurons in pattern networks connect outside of the connection channels, e.g. over or under the whole microstructure? In other words, how certain are the neural connections to follow the imposed microstructures in your case?
So there's so for that we use a technique to apply small amplitude voltage in on the on the electrodes and see how the electrode impedance varies over the spatial over the recording area. And that sort of allows us to map how well this PDMS devices are attaching to the electrode surface. And in some cases, the PDMS are not attached, and we see like a giant like detach, sorry, not giant, but like spatial detachment, a partial detachment of the film, where we see lower electrode in the areas where we should see a higher electrode impedance, but those we exclude from the experiments.
Thank you. Thank you. I think that finishes all of the questions that we have so far. Let me share screen as of the moment. So please, if you haven't followed us yet, follow us on LinkedIn and stay up to date for our next webinars or next events that we have at Muscle Buy Systems. So I hope you can see my screen right now. Yep. And next, we would like to invite you to our neural models symposium that we are organizing. Actually, also Professor Yamamoto is going to be part of this. If you want to meet him in person, ask him your questions or collaborate, this would be a good moment to do so. It's going to be on September 16th to 18th here in Zurich and Switzerland. And we are very happy to receive your abstract submissions. We are accepting posters, poster presentations, and so on. Also, short talks during the presentations. And also, I'd like to mention that we are celebrating our 10 years anniversary this year, and we are celebrating it here in Zurich. And please register here in this QR code if you would like to join us in our celebration, actually on the last day of NUMAS. So that will be a fun time to have, a tour around our office and to hear about the stories on how Muscle Buy Systems came from, the beginning up to how it is now after 10 years. So also aside from that, meet us in person in many different events around the world. We will be in Barcelona again, and in Montreal in the next few weeks. And this will be a good chance for you to also get to know more about our HD-MEAs and also to meet the team. Also, we will have our innovation showcase at ISSCR by Dr. Aline Martins about how organoids from space are being recorded by our HD-MEAs, and also from Dr. Dai Huang Tan, also recording from organoids or as the assembloids using our HD-MEAs. And finally, we also have postcards that you can get from any of these events. So if you have them, please take a picture and use the hashtag MXW postcards so that we can see them. And hopefully this will allow us to see where you are traveling around the world together with us. Thank you so much again for joining us. We are very happy to have you today again, Professor Yamamoto, very late in the evening, all live. And we really appreciate your time and also congratulations on the work. And thank you once again, everyone, for joining us. Please stay tuned for the next webinar and have a good time wherever you are. Thank you. Bye.